Welcome to Virology Live. This is session number 23. Today we are talking about HIV and AIDS, the quintessential follow-up to our previous session, of course, which was on emerging viruses. There's a wonderful, wonderful book about the origins of AIDS by Jacques Pepin. No relationship to the chef. Jacques Pepin was a doctor in Africa for many years and has insight into some of the origins. And I will quote from his book, "This tragedy was facilitated or even caused by human interventions, colonization, urbanization, and probably well-intentioned public health campaigns." And we will talk about this today in some detail, and much of which I tell you actually comes from the book. So I highly recommend it. It's not very long. You could read it very quickly, and it will enhance your understanding of the origins of viruses. The story we are going to tell today is a wide-ranging one. It begins in 1981, June 5th publication of Morbidity and Mortality Weekly Report, Pneumocystis pneumonia, Los Angeles. In the period October 1980 to May 1981, 5 young men, all active homosexuals, were treated for biopsy-confirmed Pneumocystis carinii pneumonia at 3 different hospitals in Los Angeles. 2 of the patients died. All 5 had laboratory-confirmed previous or current CMV and candidal mucosal infection. This was very unusual to see this in healthy individuals. So this caught the attention of physicians in Los Angeles. And they described these 2 cases. And they noted here, and I've highlighted, pneumocystis pneumonia in the U.S. is almost exclusively limited to severely immunosuppressed patients. So this gave an initial clue about what might be going on. And then they conclude all the above observations suggest the possibility of a cellular immune dysfunction related to a common exposure that predisposes individuals to opportunistic infections such as pneumocystosis and candidiasis. Although the role of cytomegalovirus infection in the pathogenesis of pneumocystis remains unknown, and they go on to talk about that. So this was quite a prescient observation because, as you know, we subsequently found out that these and many others were infected with a virus that we'd not seen before, and they were immunosuppressed as a consequence. Now, at the same time in New York City, at Albert Einstein Medical College in New York, not too far from where I sit at the moment, a pediatric immunologist, Dr. Ari Rubinstein, treats 5 black infants who are showing signs of severe immune deficiency, including pneumocystis pneumonia. At least 3 of these infants are the children of women who use drugs and engage in sex work. He recognizes that the children are showing signs of the same illness affecting gay men, but his diagnoses are dismissed by his colleagues, which is so typical that the prescient ones are dismissed. And I think if we'd been doing TWIV back then, we probably would have agreed with Dr. Rubinstein, but the pundits would have said otherwise. The cardiologists, who would have, of course, no business commenting on immunosuppression, would have said otherwise. So that's the beginning of AIDS. Subsequently, clusters of pneumocystis pneumonia and Kaposi's sarcoma, another sign seen in these patients, which is obvious because it manifests in skin lesions, as shown here, but it also causes internal tumors. We'll talk about this a bit later, is observed in other urban centers. And remember, these were first observed in urban centers in the U.S., but no one, well, that's not true, before I even say it. Immediately, people thought this virus was made in a lab, of course. But you'll see otherwise today. The Centers for Disease Control established the case definition for this new syndrome of Kaposi's sarcoma or opportunistic infections. That was the original case definition. In 1982, the disease was renamed AIDS, acquired immune deficiency syndrome. It was previously called GRID in a terrible indictment of narrow-minded observation, gay-related immunodeficiency, and because quickly it was shown that you didn't have to be gay to acquire it or transmit it, but it was just horrible that this would be named such and stigmatized the disease for many years. And this virus was subsequently found to be transmitted at birth and heterosexually by blood products as well. So a lot of these observations, unfortunately, ignored very early in this outbreak. Another example of politicizing public health. The virus was subsequently isolated in 1983 from the lymph node of a patient with lymphadenopathy in Paris. It resulted in the Nobel Prize in 2008 to Montagnier and Barré-Sinoussi. The work was done by Barré-Sinoussi in the laboratory. It happened to be that Montagnier was the head of lab, and he said things recently that, he should probably be stripped of his Nobel from what he's said. Anyway, we don't want to be political here. Lymphadenopathy, of course, is swollen lymph nodes. As you know, when you're infected, the local lymph nodes in the area of the infection will become activated as the T cells start to proliferate, and so the lymph node swells, and that's called lymphadenopathy. A blood test was developed for the infection in 1984, which allowed then screening of not only patients but blood supplies, because the virus was being transmitted prolificately, a lot, via the blood. But a lot of the blood supply companies denied it, to the detriment of patients who ended up getting infected via the blood. Horrible, horrible outcomes. You go back in the history of this and you can find people who head blood banks saying, no, this is not an issue, where there was evidence that there clearly were issues. And finally, electron microscopy and sequence analysis shows that this virus, human immunodeficiency virus 1, is a lentivirus, a known group of retroviruses. And of course, we have talked a bit about lentiviruses so far. They're enveloped RNA-containing viruses, positive-stranded RNA, 2 copies of the RNA. The nucleocapsid within the envelope is unusual because it is conical-shaped, I would say. And it, of course, contains also a reverse transcriptase. Now, someone in the chat is asking, which lymph nodes? Well, the lymph nodes near an infected area drain that area. So if you have an upper respiratory tract infection, typically the lymph nodes under your chin, around your throat area, will swell because they drain the respiratory mucosa. And so in some cases, patients with HIV had those lymph nodes swollen. Many of the lymph nodes, which are scattered throughout the body, and we talked about this in a previous lecture, your body's full of lymph nodes to drain different areas. You can't feel most of them. Perhaps some in your groin might be draining the intestine, but more likely there are deep intestinal lymph nodes draining that area. So the ones under your chin are really most accessible, the ones under your armpits as well. So this is a known group of retroviruses. Now here's the Retrovirid family, just to put this in perspective. Alpha retrovirus, these are the genera. Beta retrovirus, delta, epsilon, gamma, and lentivirus. Unfortunately, we are reusing these Greek letters. I don't think it's such a great idea, but no one asked me. We've talked about a number of these retroviruses before. Avian leukosis and Rous sarcoma are alpha retroviruses. Walleye dermal sarcoma virus is an epsilon retrovirus. Maloney-Murine leukemia virus, a gamma. And so HIV, one was found to be a lentivirus. And subsequently, we found HIV-2, which is also a lentivirus. Now, lentiviruses weren't new. We'd known about them before. But now we know, and to put this in more perspective, there are 2 evolutionarily distinct groups of human retroviruses. Retroviruses known to infect people. We have the HTLV, human T lymphotropic viruses, 1, 2, 3, and 4, and the immunodeficiency viruses, HIV 1 and 2. These are lentiviruses, as I've said. They are not unique to humans, and they include equine infectious anemia virus, which causes a fatal immunodeficiency of horses. We've known about that in the 1900s, early 1900s. There are bovine, feline, caprine immunodeficiency viruses, each of which cause immunodeficiency in their hosts. And we studied those for years, and that's why it was relatively quick to figure out what was going on with HIV-1, because we knew that in animals, other lentiviruses could cause immunosuppression of them. Here are the proviral diagrams of HIV-1 and 2. You should know by now what a provirus refers to. I hope you do. Don't forget some of these things. Brianne Barker showed yesterday on Twiv a photo she took at the Museum of Natural History of a poster saying the common colds may be caused by DNA-containing rhinoviruses. And I would hope that all of you taking this course would be horrified if you went to a museum and showed that. And you would immediately go to a docent and say, but that's wrong. And of course, they wouldn't know what you're talking about. Anyway, these are proviruses, which means the integrated DNA copies, because these retroviral genomes indeed do integrate after being copied from RNA into DNA. We have the LTRs on either end, and then the coding region in the middle. And here's HIV-1 and HIV-2. So these are retroviruses with complex genomes. What does that mean? Well, in addition to GAG, POL, and envelope coding regions, those are the coding regions in the retroviruses with simple genomes, there are additional open reading frames encoding a variety of accessory proteins like VIF, TAT, REV, VPU, VPR, etc., many of which we have talked about previously in terms of their function. For example, one of these encodes an antagonist of the apobex. So HIV and AIDS. AIDS stands for Acquired Immunodeficiency Syndrome. A syndrome, of course, is the occurrence together of a group or pattern of symptoms which are characteristic of a particular syndrome, right? You often call something a syndrome before you know more about it and what's causing it. And so we now know what causes AIDS, but the syndrome part remains in the name. There's no question that HIV is the agent of epidemic AIDS. And just as with every other aspect of science, there are so-called AIDS denialists who say, you've never isolated the virus, you've never seen it, you've never proven it causes AIDS, which is about as much BS as many of the things being said about COVID today is. In fact, we have inadvertently infected people with contaminated blood, proving that HIV-1 causes AIDS. There are many, many cases of otherwise healthy individuals who received a blood transfusion and subsequently developed AIDS and in fact died. So there's no question about it. You can, folks, argue all you want. You're wasting your breath. Move on to more important things. Now let's look at some of the numbers here, because we forget that there are many people infected with AIDS even today, and it continues to be a pandemic. It's infecting people globally. Even though we have ways to control it, it still is moving on. So here is the pandemic in the U.S. It's killed over 600,000 people, which is a large number, of course. It exceeds all the combat-related deaths and all the wars that we fought in the 20th century. At the moment, 1.1 million people in the U.S. are living with HIV-1, and 1 in 7 don't know it. They have not been diagnosed, and of course, they're dangerous because they can transmit it. In 2019, there were 36,000 new infections, 69% in men who have sex with men, 23% in heterosexuals, and 7% in intravenous drug users. And I have two charts here from the CDC that break this down more granularly. Here on the left, lower left, it shows the age breakdown of 2019 new infections. And so you can see most of the new infections are in the 25 to 34 age group, but other age groups are not unaffected either. They're all affected. And then on the right, different characteristics of the population. Here on the x-axis, the number of infections, and then different groups here on the left. You can see Black and African Americans, male-to-male sexual contact is responsible for most of the cases, followed by male-male, Hispanic, Latinos, and then male-male, white sexual contact. And then we have women, heterosexual contact, men, heterosexual contact, Black, African American, Hispanic, and then white women, heterosexual contact. So all groups are affected. Globally, at the moment, nearly 38 million people are living with AIDS worldwide. 38 million people, and they will do so for the rest of their lives. And in 2020, 680,000 people have died of the disease. This is down from the last time I taught this, it was over 1 million. So it's improving, but it's still a lot of people to be dying. And here's a summary of the epidemic for 2020. Again, the global number, 1.5 million acquired HIV in 2020, and 680,000 died. That's broken down into adults here. You can see women, men, and then children less than 15 years of age. Many new infections in them. This is clearly a global disease. I'll show you the map of the world in a moment. 4,600 new infections a day, 190 per hour. As we're sitting here for the next 2 hours, we're going to have 380 new infections. So this is a substantial pandemic still. Here it's broken down by WHO global regions, right? The WHO breaks down the world into the East and Southern Africa, West and Central Africa, Asia Pacific, etc. And so you can see the vast majority of cases shown by the number of human icons. Eastern and Southern Africa, 20 million. Western, Central Africa, Asia Pacific, also quite high numbers, and other areas as well. But there, the greatest burden is in East and Southern Africa. What about population groups globally, not just in the U.S.? So the most shown in this pie chart is the remaining population, but we have 20 sex workers, we have 11%, people who inject drugs, 9%, 23%, a large fraction, men who have sex with men, transgenders, 2%, clients of sex workers, 20%, and the largest part in the remaining population where we don't have an identification. And finally, the global picture of infections in 2020, new infections in 2020 according to region, but also the change since 2010. The decrease in infections shown by the arrow here. So here, for example, we have had 67,000 new infections in Western and Central Europe and North America. These are not really the WHO regions any longer. And that's gone down 37% in the last 10 years or so. Caribbean's gone down 28%. Latin America, it stayed the same. Western and Central Africa, it's decreased. East and Southern Africa decreased. Middle East, however, has gone up. Europe and Central Asia has gone up. Asia Pacific has gone down. So some places are doing well, others are not doing so well. And as we talked about last time, we can control the disease with triple drug therapy. It has certainly slowed the pandemic in countries that have money, higher income countries. But it's not as widely available as it should be in other countries. And that's shown here. This is antiretroviral coverage over time. And, we started out very low when the triple therapy was first devised, and we've gotten to 62% of the people who need it, but that's not enough. We need to get over 90%, obviously. So there's still issues at getting drugs to the people who need them, just like getting vaccines is always an issue in getting them to people who need them. So let's take a break here and look at some questions. When is there a version of Virology live in Italian coming? Let me start my timer here before I forget. I don't think I could teach an entire course in Italian. My Italian is not good enough. However, if I lived in Italy for, say, 3 months, then I could do it, but I'm not likely to do that anytime soon, although I would love to. It's a lovely place. Were the cases discovered in L.A. The first known cases in the world? Well, there were cases in many urban centers, but they weren't recognized. They weren't recognized to be a commonality. It was only in large centers like New York and L.A. and San Francisco, Chicago, that physicians were reluctant to say this is something. Only a few, and the morbidity mortality report was unusual in that they said, yeah, there's something going on here, and most physicians didn't want to believe that. So there were certainly other cases going on, and as you'll see later, they've been happening since 1920, and we've seen them, but we didn't put it together. Difference between HIV 1 and 2 we'll talk about in a bit. I'm going to go over that in some detail. Yeah. Yes, as Vanity has said. Wasn't that the case that some nations in Africa resist vaccines? Yes, of course. There are, African nations that are not democratic. They are run by dictators who have their own beliefs. They don't want you to use condoms. They don't want you to use drugs. They're Western products. They're going to do things. Yeah, it's always an issue. Unfortunately, taking care of people's health is not straightforward. Has the young adult age group been especially vulnerable? We have these statistics for every year, and there is a chart answering your question. I'm not going to say yes because I don't have it at hand, but if you look at some of the links on my slides and go to those websites, they have wonderful charts, tons of them, and it'll answer your question. Why is it higher among African Americans? Well, I don't know the answer to that question. I think it's probably a factor in healthcare, getting healthcare early and getting right counseling and so forth. But I don't know offhand. Ryan White was an American teenager with hemophilia who became a national poster child, died in 1990. Yes, people who needed blood products got infected because the blood companies didn't believe that there was much HIV in their blood. They were just idiots, downright idiots, looking for profit over anything else. Are men more in risk to get infected than women because of similar differences that we see with COVID? So it's not just genetics. We're going to talk about that in a bit. But, most of the sexual transmission, if it's intravenous drug use, there's no gender difference. There's no sex difference. It's sex in itself that is different. So that, insertive versus receptive and all that. We'll talk about that in a bit. I want to work in a virology lab. I have a fear of mice. Well, you don't have to work in mice in every lab. So there are many, many labs that don't work on mice. So just find one that doesn't. I've been working with mice for 40 years. I have no problem. I like mice. Professor here in Finland told us that Finns are much more reduced risk to get HIV. I don't know what the basis for that is. As you know, we've already said the people with deletion of CCR5 gene are resistant to infection, but they're only 4 to 16% of the world's population. So, there may be some of those individuals, but other than that, I don't know. Thank you for your contributions, 8440K. So 8440K refers to a Coursera course I did years ago where you could get a certificate. I haven't put that back up, but I've always wanted to. And I really need to have some help to do that because it involves breaking all these sessions into short videos and making questions, and it's a lot of work. And I think it's worth doing that because we had huge, huge numbers of people registering. So yes, I will do that at some point. Ian, parli italiano, io non parlo, io sono grasso e brutto, ma tu sei bellissimo. That's not true. I'm sure you're not fat and ugly. I speak a little bit, parlo un poco, ma non molto. Do we specifically know why HIV vaccines don't work? We're going to talk about that a bit later. The answer is no. Has any natural resistance been observed? Some. We've talked about one, the deletion of CCR5, and there are other people who are not completely resistant, but more resistant, which we'll talk about. Yeah. Why are men who have sex with men still restricted in regards to donating blood when we know that a person could have HIV/AIDS? Well, they are a high-risk group, right? They are not the only risk group, but they're among the highest, and so that's, I think, the logic in it. Yes, Les, they did have another correlation. They actually had CD8, CD4 ratios. A scientist in San Francisco actually came out with that and said, look, you can test blood for the ratio and get an idea, and the blood banks didn't want to do it. Nope. Is there a good mouse model? So there are humanized mice where you replace the immune system with the human immune system, and that is somewhat useful, yes. Is there any example of recombination? Yes, not with a non-retrovirus, but among HIVs, certainly. But no, not with others. Does SIV cause AIDS in non-human primates? Yes, absolutely. Simian immunodeficiency virus. We're going to talk about that a bit. Why is the CCR5 in Europe? Wouldn't it make more sense in Africa? But it arose actually long before the HIV pandemic began, many, many decades before. So there was some other selection pressure for CCR5 deletion, and we don't know what it is. Could you give me a tip to pass my microbiology exam? All right, look. Study. Don't get distracted by the world. The world is easily distracting you. But just get into it. First, you tell yourself this is the coolest stuff on the planet. There's nothing better. And sit down. I don't know how you study. Everybody studies differently. Take notes, watch videos. Taking notes is really important. Take practice exams. Go online. You can find so many resources online now. And do it for a long time. Don't cram the night before because you're not going to get it. How long does HIV remain viable and infectious? Not very long. That's why it's not transmitted except by intravenous drug use and sex, because it doesn't do well in surfaces and aerosols. And we'll get back to that in a minute. So the Delta 32 makes you resistant to viruses that use CCR5. And there are some subset that use CXCR4. However, most of the transmission is by R5 binding viruses. And so that's why it's effective. But if you took a CCR5 deleted person and injected them with a CXCR4 utilizing virus, they would be infected. Well, certainly here in the U.S., blood is tested for HIV, and it should be elsewhere, of course. Yeah. Blood donation is also rejected if the intravenous drug use history by donor or sexual partner. Yeah, I always like taking notes. I mean, not just verbatim copying, but translating the information into your hand, and that helps to learn it, for sure. I think that's a known learning mechanism. I don't know if it works for everyone, but it certainly works for me. I don't know most of the characters. I know the husband and wife. Intravenous drug use transfers blood. Of course it does. That's how you transfer the virus, in the blood. There's contaminated needles. Problem is, intravenous drug users, if they all used a brand new disposable syringe, it wouldn't be an issue. But if they share them, that's the issue, because there's a little bit of blood left in the syringe. All right, so we've now gone through 10 minutes. That's cool. Now we can go back to slides. Here we go. All right, so we have triple therapy, but there's no cure. You cannot get rid of virus from an infected individual. You can keep their disease down very nicely, but the virus is always there. If they stop taking drugs, the virus begins to reproduce. So that's a problem. There's no vaccine. We've tried a lot of vaccines. Every one of the trials has failed. So you cannot block the primary infection. I do think a vaccine would be terrific, but we don't have one. I'm not sure we're going to have one for a while. So you can't stop taking antiviral drugs. There are reservoirs, latently infected hematopoietic progenitor cells in the bone marrow, in the lymphoid organs that give rise, where the viral replication is suppressed, periodically becomes activated, and new virus is made. We also still get drug-resistant viruses, as we talked about, and drugs are expensive. So we still don't have a solution, and that's why, in part, the pandemic continues. Let's talk about the origins of HIV. This did not come from a lab. It took a long time to figure out where it came from, and until then, people said it came from a lab. And this is in part why I am so upset about lab leak hypotheses, because they are founded in nothing, just like the lab leak HIV hypothesis was founded in nothing. And I object to nothing. I want data as a scientist. And maybe people who are not scientists don't understand that. They want to rely on hearsay. So the first studies in Africa, after we discovered HIV elsewhere, they went and they used the blood test and they say, who's positive? First studies in Africa, in Zaire and Rwanda. The names of these countries have changed over the years, right? So this Rwanda, Zaire, showed that AIDS was common in Kinshasa and Kigali, where 90% of sex workers were infected. So serological surveys, 90% of sex workers were infected in those areas. We had no idea. No idea. So then we started testing archival samples, samples of tissue and blood that had been frozen for many years. They were stored appropriately, and they showed you could look for the genome, that the virus was present in the 60s and 70s in several locations in Central Africa. So here we're looking at Central Africa here, but not in Western Africa and not in Eastern Africa. Yeah, it's interesting, right? It's getting some hint about its origins then. Two of the earliest samples that we have initially was ZR59 from a DRC male, here DRC, DR Congo. 1959 was found to be positive for HIV-1 in 1998. And then a lymph node sample from a DRC female, 1960, both in the same country in Central Africa, positive for HIV. So this virus was around in 1959 and 1960. No question about that. So much for made in a U.S. government lab. These two viruses differ by 12%. That's a lot. Genome sequence. And so these have been around already a while because these two have diversified. So there's no question that HIV was present by 5960 in Kinshasa. At the time, it was called Leopoldville, right, the capital of the Congo. So what's the source? Where did it come from? Well, in the 80s, SIV was first isolated. So a number of years after HIV, we started looking in chimpanzees, non-human primates, and asking, are there related viruses? And indeed there are. There is a virus called SIV, Simian Immunodeficiency Virus, or CPZ for chimpanzee. You'll see why we have a specific name for that in a moment. And so the idea arose that perhaps this was the source of HIV. And Beatrice Hahn here, a virologist at the University of Pennsylvania, that's a photo of her in my lab. This is when the wall of polio was a freestanding art piece in the laboratory. It fell. I used to take pictures of people in front of it, and it fell several times, so I moved it to my office. She directed a study where over 7,000 chimpanzee fecal samples were collected from 90 field sites throughout Central Africa. It turns out the virus can be detected in the feces. It can also be detected in urine. And so the sampling is quite interesting, and I've spoken with her. I've interviewed her before. You can find that interview. I think that's it right there. That's the link. The chimps sleep up in the trees, and in the morning when they wake up, they pee, and the workers can stand under there with containers and collect their pee, and they can see who it is. But they go back and they do mitochondrial DNA sequencing, and they can identify which chimp it is. And then they can do PCR and look for SIV. And they confirmed that chimps are indeed the natural reservoir of SIV-CPZ. But it's not in every chimp. So here on the left is a map of Central Africa with all of the chimp colonies that they sample. So chimps live in little communities. And they don't interact very much with each other. And especially if there's a river in between them, they never cross it. And so here, these are all the different kinds of chimps. Pan-troglodytes is the species, and then we have pan-troglodytes, troglodytes, PTT. We have PT varus, PT elioti, PT schweinfurthi, and P. peniscus, which is the bonobos. They're not troglodytes, but they are chimpanzees, bonobos. And only PTT and PTS harbor SIV-CPZ. And that is shown by these yellow-colored circles. These are all the colonies she sampled, and only these yellow ones harbored SIV-CPZ. SIV is transmitted among chimps by sex. It's also transmitted mother to child during birth, and possibly during aggression. Chimps do fight, and they can bleed and infect each other that way. The estimated transmission probability per coital act in chimps is 0.008 to 0.0015, which is similar to that in humans, 0.0011. That's the probability per coital act that you will transmit HIV. It doesn't mean that you need to have sex over 1,000 times to transmit. It could be transmitted that first time, but it's a frequency. So don't think, hey, I'm okay for a lot of sex, I'm not going to get it, unprotected sex. No, it doesn't work that way. Furthermore, the SIV-CPZ is pathogenic in chimps. It causes a disease similar to AIDS and immunosuppression, which initially wasn't thought to be the case. It was only subsequently. In fact, the certain individuals, Jane Goodall, for example, who work with chimp colonies, noted that some chimps would die. For example, Beatrice Hahn had a relationship with Jane Goodall, and she would call her and say, "Oh, I need some more blood from this one. Oh, that one died." And from that kind of interaction, they realized that it was killing the chimps. And then, of course, the genome sequence then shows that SIV is the ancestor of HIV-1. So here are gene sequences of the GAG and the PAL and the envelope genes for a variety of isolates. So we have SIVs on here. We have SIV-CPZ from PTT, that's pan-troglodytes, troglodytes. And you can see that those sequences cluster with HIV-1 M. M is the main group of HIV in humans. We'll talk more about that in a moment. There's also an N group of HIV, which clusters with CPZ, PTT. And then we have a whole bunch of other PTT sequences here. And then we have gorilla isolates. They're SIV-GOR, gorilla isolates of SIV. And those are they have a common ancestor with HIV-P group. And then we also have an HIV-O group, which shares an ancestor with another gorilla isolate. So these share common ancestors. So HIV, obviously, the contemporary SIV-CPZ cannot be the ancestor of HIV-M because they're co-circulating, right? Rather, they share an ancestor and obviously are highly related. And this goes for both the PAL and the envelope gene as well. So there's no question that the ancestor of HIV, all the different groups, which we'll talk about in a moment, is SIV-CPZ or SIV-GOR. So what's the story? Now, here's our catalog of non-human primates and old world monkeys. So here on the left are old world monkeys of different species, Mona, red-capped mangabe, mandrills, vervets. If you've ever heard of African green monkey kidney cells, they're actually vervet monkeys is the correct name. The kidney cells are made from these animals, Sykes monkeys, sooty mangabes. So old world monkeys, which live, of course, in the old world, like Africa, are themselves infected with their own SIV. So each one has a species-specific SIV. So the Mona monkey has SIV Mon. The vervet has SIV Ver, etc. These monkeys have been infected with these SIVs for thousands and thousands of years, and they do not get immunosuppressed. These old world monkeys are absolutely okay with having SIV infection. And a few hundred years ago, they transferred SIV to chimps. And in fact, the chimpanzee SIV CPZ is actually recombinant between the SIV Mona and the SIV red-capped mangabe. So somewhere hundreds of years ago, these two viruses were transferred to a chimp. They recombined and became SIV CPZ. How did that happen? Well, chimps actually eat meat sometimes. So they could have eaten an old world monkey and acquired their SIV. And it is SIV CPZ that was subsequently transferred to humans in two separate spillovers to form the M and N groups of HIV. The gorilla SIV Gore, came from a spillover from chimps. SIV CPZ spilled over into gorillas, became SIV Gore, which then spilled over into people to form the P and O groups of HIV in humans. So it's a rather complex history, but it all starts with old world monkey SIV, goes to chimps and then gorillas and then to humans. So we have four separate spillovers from either chimps or gorillas to lead to the four lineages of HIV that we see in people today. And there probably have been many, many spillovers, but we just don't recognize them. Now, when did SIV infect humans? This is we can figure this out for the M and the O groups. And again, there are four separate crossover events, 2 from chimps, 2 from gorillas. M and O probably spilled over in the first 3 decades of the 20th century. That's a long time, right? 30 years. So I put it at 1920, roughly, between 1900 and 1930. N and P probably more recently, but there are so few cases we don't have enough data. So again, the M and the O, chimp and gorilla, 1920s. The N and P more recently, but we don't have enough data to sort that out. Now the question is, how did this virus infect humans? And the best idea, I think it's an idea. It's not a hypothesis because there are no data to specify this, but it is the cut hunter hypothesis, which is someone was hunting bushmeat, which means you go in the woods and you hunt typically a chimp. Gorillas are very difficult to hunt. They're very elusive. But you can hunt chimps, and it's known to have been done for many years throughout Central Africa. And so the hunter captures a chimp, starts to butcher it, cuts him or herself, gets exposed to blood from the chimp, and then gets infected with SIV. And in that book, The Origin of AIDS, they go through these calculations. They suggest that in 1921, the number of people infected with SIV CPZ was less than 10, but one of them spread and multiplied and became the main group of HIV. And such cross-species infections probably have happened many times before and after, and this is the main one that spread. Why? Well, because we helped it. Here's where humans come into the story. Well, of course, they're already in the story because we're out there hunting chimps. But you look at the development of Africa over the years, and you see that the increased population growth, especially in major cities, is what drove HIV through the population. Now, here's Leopoldville, Kinshasa, down here. It's the most dynamic city in the region, attracted large numbers of migrants and traders. You can see Kinshasa, 1883, a few homes here and there. By 1961, extensive development around the river there. And then Here you should go from top to bottom. 96, a little more developed. 1955 and 1961, huge urban centers. And so what's happening here is that people are moving from the countryside to these big cities. The cities, of course, then have brothels because the workers who are being imported, which I'll talk about in a moment, need brothels, I suppose. So this cut hunter of ours might have traveled to one of these cities and visited a brothel, then an STD clinic to get treatment for the sexual disease that he acquired. And those would use non-sterile syringes. And they would propagate the virus to others, just like hepatitis C virus was propagated in Egypt. It was propagated by sex. Some women had 1,000 clients a year, and they're propagating the virus. And that amplifies the virus throughout Africa. And here's the population growth of the region from 1880 to 1970. And you can see these centers have low populations. And the M group, time to most recent ancestor, spans this large number of years from 1880 or so to 1920. So it's any time in here that it could have spilled over from a chimp to humans. But you see the population is then growing in these cities, an explosive growth in the 40s and 50s, and this is spreading the virus as the population grows. Now, you will see in a moment that the virus entered the U.S. through Haiti, and that is because the Belgian Congo in the 60s, they were taken over by Belgium before the 60s. And in the 60s, there was a revolt, and they said, get out of here. We want our country back. So the Belgians left. But unfortunately, most of the doctors were Belgian. So then the Congo needed doctors. So they reached out to Haiti because people speak French in Haiti and in the Congo. So people came from Haiti to work as physicians in the Congo. And then they got HIV and brought it back to Haiti. And so colonization of Africa is a big factor here, European colonization. Look at Africa being divided up among the European countries. If you want a piece of Africa, and they move in, they establish large population centers, they move males for labor, they move them from the countryside. Remember, Africa, colonial Africa, before colonial Africa, is just a bunch of little chiefdoms with small numbers of people, and then the European countries take over. And this is not indicting you because everybody does this, but then they say, we're going to build large cities, we're going to move the males in, take them away from their families, move them in to build roads and bridges and railroads. And of course, the brothels come into play there, large-scale prostitution. And then the European countries also say, we need to take care of the health of these people. So they establish colonial medicine, healthcare centers, and they inject people with the virus because they're using non-sterile syringes. As we mentioned in Egypt, the use of non-sterile syringes spread hepatitis C virus to millions. So this likely resulted in large-scale amplification of HIV-1, largely in Africa for many years. And, of course, it was exported elsewhere. And there was certainly immunodeficiency, but we didn't notice it. Now, here's a wonderful figure from a paper published not too many years ago by Michael Waraby and his group. We just did a paper of Michael's on TWIV about the early origins of SARS-CoV-2. But he looked at samples of genomes more extensive than had been looked at before. So he had constructed a phylogenetic tree of patient samples that he recovered in the 70s in New York, in 78 in San Francisco, Haitian samples. And he deduced that, again, the virus originated in Central Africa. It moved to Haiti in the 60s when the Haitian doctors went back and forth, went from Haiti. Haiti was a vacation destination, and so it moved to New York then, and you can see late 60s, early 70s, moves to Georgia, and you can see these green samples are from patients in Georgia, and also moved to California, where we can see these red samples. And I don't know if you know, but there was a famous patient zero, Gaetan Dugas, who was supposedly started most of these chains. This isn't true because his genome sequence is just buried in here with all the others. He wasn't anything special in terms of origins. He just infected a lot of people. HIV-2 was first isolated in Guinea-Bissau, Western Africa here. Has about 30 to 40% genome identity with HIV-1. And it is restricted primarily to Western Africa. It is less virulent than HIV-1. In fact, most infections do not progress to immunosuppression. It is less transmissible, and there is no mother-to-infant spread. This virus is a crossover from the sooty mangabey directly into humans. So SIV, SMM spillover into humans results in HIV-2. And this has happened at least 8 times. There are 8 distinct lineages of HIV-2, A through H, each of which arose from a separate spillover from sooty mangabeys to humans. This was also introduced into macaques in the U.S. and other countries studying these. They would inject macaques with serum from these mangabeys and inadvertently infected the macaques, which gave rise to a new lineage of SIV called SIV-MAC. Now, HIV-1 undergoes extensive diversity because it's a retrovirus. It has a high genome mutation frequency and no error correction. And so we now have 4 groups based on sequence alignment, group M, N, O, and P of HIV-1 only. The group M stands for Main. 99% of all HIV infections are group M. Group O is called O because it stands for outlier. It's less than 1%, and it is limited to Cameroon, Gabon, and neighboring countries. Group N, there have been only 13 cases found so far in Cameroon, and Group P, only 2 cases in Cameroon. Of course, Cameroon is where we think the original spillovers of HIV-1 from chimps and gorillas occurred. But remember, each of these 4 groups is an independent transmission event of SIV, CPZ, or gorilla to humans, from chimps or gorillas to humans, not old world monkeys, chimps or gorillas. HIV-2 are spillovers from old world monkeys. Now, HIV-1 further diversifies into subtypes. Why is that? Infecting millions and millions of people, more hosts, more mutation, more diversity. Sound familiar? Well, that's what's happened with SARS-CoV-2. It's nothing special. So now we have 9 subtypes, A through K, of group M, because that's the most numerous. We also have recombinants. They're called CRFs, or circulating recombinant forms, because high-risk individuals, like prostitutes, are multiply infected, and the viruses recombine, and if the recombinant has a higher fitness, it will spread. Not because it's more transmissible, it's more fit. And the recombinants, 48 of those so far. There's no clear-cut difference between these subsets in virulence. There's some observation that people infected with D die faster, but I think those kinds of observations are always subject to error. Subtype C shedding is higher, perhaps associated with higher female-to-male transmission. This is extensively spread in Africa. I'm not sure any of that matters. I think that when a subtype arises, wherever it is, it predominates. And in fact, the subtype that's introduced into a region becomes the founder. So some subtypes are associated in specific locations with modes of transmission, and that's due to a founder effect. A subtype will predominate in the at-risk group in which it's introduced. So, for example, subtype B is found in 96% of white homosexuals in South Africa. It was imported from the U.S., where it predominates, and it was obviously imported in a white homosexual who then went to others in South Africa, and they're spread amongst each other. That's why they predominate in certain groups of individuals. For black heterosexuals, subtype C accounts for 81% of infections. So whatever subtype initially goes into that group, and they're different groups of people, that is what predominates. It has no biological relevance, in my opinion. This is a map of all the subtypes globally. You can see the pie chart. Here are the subtypes here colored, and they're circulating recombinant forms. And you can see subtype C predominates in South Africa. As I've just told you, it predominates in some other countries. Here in the U.S., we have predominantly subtype B. Obviously, that was the subtype that was introduced many years ago, and it's simply spread. And in other countries, Central Africa has the most diversity because it's been there the longest. It spilled over there from chimps and gorillas in the 1920s, 1900s, and it has had the longest time to diversify. And the global population, the fraction of subtypes is shown here. Let's take a question break and see what you've been thinking about. You may ask, why does HIV get its own session? Well, because it is a pandemic and we've been studying it for ages. It's a big deal, and we understand a lot about it. Okay. How did PCR help diagnosis of HIV from the 80s? Well, initially it wasn't PCR. It was initially antigen and antibody tests were developed, but then PCR came online in the 80s, and so that was developed, and sequencing, genome sequencing as well. High throughput was developed in the 90s, and that's why we have a lot of information on subtypes now. Why vaccines haven't worked? I'm sorry, I didn't highlight that. Sorry, there you go. There's that question. Why haven't vaccines worked? Very complicated issue. We'll touch on it in a bit. 20 years ago, in my locale, AIDS became quite common among retirees living in retirement communities. 20 years before that, AIDS was considered to be a disease. Yeah, so this is how things have changed. There's no reason to think any virus is a disease of any particular population. It's just a bias. This morning, University of Kansas podcast, Dave kudos for 844 and recommended everyone watch regularly. Very awesome. Had to share. That's great. I'm really glad to hear that. We are slowly but surely making inroads, right? Even if we get 10, it's good. Lynn, thank you for your contribution. Spent my last semester in nursing school in 1990 caring for patients with AIDS at a Washington, D.C. hospitals. It was tough. So many advances in treatment. Yeah, it was tough because most of them would die. There was nothing to do for them. Yes, none other than Tony Fauci took some risk in his career to work on HIV in the early 90s. That's absolutely correct. Can HIV be detected in human feces? Sure. As you will see, it is a way that the virus gets in. It reproduces extensively in the lymphoid tissues in the intestines. So absolutely present there. And in fact, can reach the intestine from, even if you get IV drug inoculation of HIV, it will make its way to lymphoid organs, which are abundant in the intestines. Thank you, Rowan, for your support. Really appreciate it. Very cool interview with Beatrice Hahn. I was sitting in class in the early 80s in Berlin and remember our professor constantly sprinkling in remarks about Fort Detrick as the origin. Yes. Yeah, right. I have read that there are blood samples from the 20s with HIV. I'm not aware of them. I'd love to have a link to that. Chimps fight viciously. Oh, I forgot to turn on the darn timer. I'm sorry. Chimps fight viciously. They can even become momentarily cannibalistic. Nothing shows aggression and asserts dominance like ripping your dead adversary's arm off and taking a bite out of it. Whoa, thank you for that. Yeah, that's what I understand as well. SIV CPZ transmission probability seems off. Well, that's from a reference. So why don't you complain to the people who wrote the reference? Okay. Now I don't have the link there, which is not a good thing, but I'll try and find it and take a look at it. You have to listen to your language ruined for so many people trying to talk. I'm not sure, but Italians are broad-minded, so lectures in Italian will probably work. I don't think I know all the words. That's the problem. There must be more spillovers happening now. Is it being monitored? No, it's not. I mean, the state of Central Africa is not good. And no, I'm sure there are, yes. But, there are much fewer chimps and gorillas. because most of them have been wiped out. So I'm not sure how frequently it is. For me, the SIV to SIV gore is a little harder to understand. Gorillas die in the woods. Maybe chimps will eat them. I don't know. I'm not a non-human primate biologist. That's what the virologists think. A journalist who covered HIV gave me a copy of Jacob and Lily Siegel's notorious manuscript, Sciencey Nonsense. It's nonsense. Throw it away. Paul Farmer writes well about HIV and AIDS. Okay, I'm not aware of that. In 1982, Steph, thank you for moderating today. The ICU, we were taught that the demographic were 3 H's, homosexuals, Haitians, and hemophiliacs. It's just such a bias. It's horrible. Of course, in retrospect, we see it's horrible. But we should have been more open-minded. You know, the president of the U.S. would not even mention, and that was Ronald Reagan, he would not mention HIV/AIDS for years until his friend Rock Hudson died of it. Then he realized it was a thing. And that's not good. Can you imagine a president not acknowledging a disease? You wonder why I get upset about this stuff. Is Group M for main common? No, virologists are cowboys. They do what they want. There is a naming convention authority, but I'm not sure that back then people listened to them. Do you know if they tried HIV-2 for an HIV-1 vaccine? I doubt that would work. They're too distinct. You wrote there's evidence of HIV-2 being less virulent and transmissible. This is clear from the data. Well, I think it's clear from population behavior. It doesn't spread outside of Western Africa. And if it really were transmissible, intrinsically transmissible, it would. And people who have it there do not progress to immunodeficiency. So I think that's good enough evidence, yeah. Could HIV-1 and 2 coexist? Yes, they could. They're utilizing the same receptors, but they could. What keeps HIV-1 and 2 from recombining? I think the genome sequences are too distinct. What did we learn about viruses from studying HIV? Well, hopefully today's lecture will tell you that, but we've learned about how immunosuppression can occur. We can learn about how latent reservoirs are established. We've learned about how they're reactivated. We've learned about various aspects of drug development and resistance. And we've learned that not all vaccines can be made. We are spoiled with COVID vaccines. People have been working on HIV vaccines from the beginning, and we can't do it. Thank you for pointing out that distribution of subtypes is more about social structure than biology. Yes, that's absolutely right. You say founder effect is in a particular COVID variant predominance due to founder effect as well. I think it's likely part of it is, I think the D614G variant was in the right place at the right time. And, the lab studies have shown very minimal differences in infectability by D614G. And the authors of the study admit they can't distinguish if it's a founder effect or a fitness effect. The subsequent variants, it's hard to say, but it's certainly an improvement in fitness that's driving them through the population. What are the criteria for doctors to test patients for HIV? So it can initially present as flu-like symptoms. I know from talking to students that they'll have a lingering illness that doesn't go away with swollen lymph nodes, and they get tested. And that's typically one. Or you may have signs of immunosuppression like candida, oral candida, or persistent fatigue and flu-like symptoms, and that drives you to get tested. I have not worked with HIV, no. A person who gets HIV-2 first is infected by one, will they get AIDS? I don't know the answer to that. Is it possible to block doors? I would suspect not, because not every CD4 T cell is going to be infected. Sanusi was a laboratory, was a staff scientist, not a postdoc. She was a member of the faculty. I think we've answered Trevor's question. Same receptors. They, as far as I know, they don't recombine. Sorry. What makes HIV dormant cells initiate transcription? Well, it's random, frankly. These cells that are latently infected and suppressed, they're suppressed by epigenetic mechanisms like chromatin modification. And these just randomly get reversed in a stem cell. The stem cell may have to start to differentiate, and then the provirus is activated. Though we know that the virus binds these receptors, is there any reason that it's more fragile and would not survive outside? Well, there are obviously reasons. We don't really understand them. It's hard to know, right? Because you don't make more stable viruses and then study the differences. People just don't do that. But this virus is spread by close contact, by drug use or sex. So it doesn't have to be stable. It was a virus that in chimps wasn't stable. And in humans, it's not, because it doesn't need to be. It can be spread successfully without being stable. If this were a respiratory virus, it couldn't do that, of course. In what percentage do antivirals fail to treat? Talking about triple therapy, it's very low, in the low percentiles, I think, and it's usually because of resistance. But if you test, you would detect that ahead of time. I remember GRID being discussed at UCL in 1978. It was said to be genetic, and nobody who thought it might be a retroviruses could get a grant to study it. Yeah, there's always this damned bias all the time. It's unbelievable. If vaccines were not invented, today the world would be different with well-developed drugs. I don't know. Maybe not. Drugs are not the solution, right? Because they cannot, in most cases, take care of you once you're infected, except we know for, vaccines, I just said what vaccines do. Drugs are not the only solution because their effect is temporary, right? So I can't imagine a world where we wouldn't have vaccines. Is HIV the most studied virus ever? Yes. And SARS-CoV-2 is not yet, because it's been too short a time. HIV has been studied for many years. I mean, it could be at some point it'll be exceeded by SARS-CoV-2, but who knows? All right, let's stop here because we need to move on. We can take these questions later. We will go back to our slides. All right, transmission of HIV, sex, intravenous drug use at birth, R0, 2 to 5, not respiratory, alimentary, or vector routes. Here are some pie charts of transmissions in different areas. So here, Western and Central Europe and North America. Men having sex with men, 57%. Sex workers, 26%. Injecting drug users, 7%. All others, 10%. But look at this, Eastern Europe and Central Asia, mostly injecting drug users, followed by sex workers, and men having sex with men far less. And in all cases, mother to child at birth, about 5%. The infectivity is severely reduced by air, drying, 99% infectivity lost in a day. It's lost by heating. It is lost by bleach or alcohol, extremes of pH. So to answer someone who asked about this earlier, that's why this is a sexually transmitted intravenous drug use transmitted virus, because that keeps it away from all those extremes. Once it gets outside the body, it's dead in the water, but it doesn't have to be. And apparently, it's never evolved. It's never been selected to be more stable outside. Perhaps it's not structurally compatible with it, but certainly the close contact transmission works. Here are some numbers for risk of transmission. Different modes of transmission and infection risk per 100,000. Let's see, I have a text, and I'm going to see if it's from one of you moderators saying something's wrong. No, okay, good. So infection risk per 10,000 exposures. So sexual transmission, receptive anal sex, 138 per 10,000. Insertive anal sex, 11. So you see the mode of sex it influences the transmission. So in other words, if you're the receptive person here, this is the infection risk. If you're inserting, this is your risk. All right. Receptive. Let me fix that typo. That is not good. I don't know why that's been there for 17 years. Now I made it worse. Receptive. Insertive is not an E at the end. I don't even know why it's telling me it's wrong. It's not unassertive. I shouldn't have done that. I'm sorry. Receptive penile vaginal sex. So this is anal. This is vaginal. Are both similar, insertive and receptive, and lower than receptive anal. Okay, parenteral. Transfusion of infected blood is almost 100%. You put a virus in you, and that's it. It's going to infect you. Needle sharing, 63 out of 10,000. Needle stick, 23. And if you have a needle stick, which may happen in a healthcare setting, you accidentally stick yourself with a contaminated needle. If you get treated with post-exposure prophylaxis, in this case just AZT, this study was done a long time ago, reduces it to 1. So that's why if you get a needle stick and you're at risk for contracting HIV from that, we recommend you do a course of post-exposure prophylaxis, which today won't be AZT, it'll be something else, but it's very effective at preventing infection. Mother to infant, no drug treatment, 2260 out of 10,000. And with AZT treatment of the mother just before birth, less than 1,000. So again, treating the mother to reduce the viral load is very effective. All right, now let's talk a little bit about co-receptors for the virus. We said before that the major co-receptor, of course, is the beta chemokine receptor CCR5. Envelopes that bind this co-receptor are known as R5 tropic. In some patients, usually latent disease and along with breakdown of the immune system, HIV particles bearing envelopes that can enter cells via CCR4 arise. These are called X4 tropic. And the reason for the switch is not known, but it follows extensive evolution of the virus population and the emergence of dual tropic intermediates, in fact. So it can use both, and then it goes to R4. The prevalence of these also depends on the clade of the virus that is being studied. Now, some of these envelopes can mediate entry into macrophages. That's what we call macrophage or M-tropic viruses. The number of CD4 molecules on macrophages compared to lymphocytes, T cells, is much lower. And so these infections are not very efficient because the virus needs high density of CD4 to infect efficiently. But during the course of infection, viruses encoding envelopes that can efficiently enter cells with low CD4, like macrophages, do evolve. And these M-tropic viruses are typically found in the central nervous system in patients later in disease. As we've talked about before, people with a mutation in the gene encoding CCR5 delta 32 protects against the R5 tropic viruses, which are the main ones that are transmitted. This deletion is found in 4 to 16% of people of European descent, and that was taken advantage of in stem cell therapy of both the German and London AIDS patients who were given a bone marrow transplant. Their bone marrows were ablated. They were replaced with a transplant from a CCR5 null person, and they were cured of AIDS. So this has led to the idea that we could possibly disrupt CCR5 with CRISPR-Cas9 approaches to do a gene therapy approach to perhaps curing people of AIDS. Bone marrow transplant is not the answer because it's quite costly and has a high risk associated with that. But perhaps CRISPRing out the gene would work. As you know, there have been some recent cases of individuals with spontaneous cures. They're HIV infected, it's quite clear, and then suddenly they became HIV negative. These are not CCR5 null people. I have a suspicion that they're not actually free of virus. It's just so low you can't find it, but we'll see. And they have no idea why this happened. Now, primary HIV infection, virus could enter you as particles or within cells. It's very hard to know the difference because when you give an animal, most of these studies are done in macaques, infecting them with SIV, and the assumption is there are similar things going on. If you give them infected cells, there's always virus particles present, so you never know what is initiating the infection. And most of those SIV studies are done by intravaginally inoculating the macaques with either virus or cells, and both can initiate infection. Virus can initiate infection. Free and virus-infected cells can initiate an infection. So somehow these viruses have to cross the mucosal barrier. There is most likely abrasion of the barrier in both vaginal and anal sex that allows virus particles to cross the mucosal barrier. That's well known for many pathogens. The virus particles are likely initially taken up by dendritic cells. There's a molecule on their surface that will attach to the virus particles. And as you know, dendritic cells, once they encounter an antigen, will then go into the lymph node and present it. And there in the lymph node, of course, are plenty of T cells for the virus to begin reproducing in. It's almost like a Trojan horse bringing the virus to the local lymph node. Of course, the virus may encounter T cells in the submucosal tissues. It can infect them as well. The infection of all these lymphocytes results in high levels of viremia dissemination to other lymphoid tissues throughout the body, and there's a short period of symptomatology, which we'll see in a moment, and then the virus levels are regulated by the immune response, and you reach what we call a set point after about 6 months or so. So here's a diagram of the progression of HIV infection. At the top, we have a nice graph showing RNA levels, copies per mil of plasma, over time. And looking at years here, we're going from weeks to 12 years because it may take that long to kill the individual. So we have that initial acute phase where we have a high level of viremia. This is virus in the blood now. It's measuring it very, very easily. And it's produced by infection of all the T cells. And you may have symptoms, swollen lymph nodes, fever, diarrhea. The CD4 cells decline temporarily. That's shown in the bottom panel. So the bottom panel is measuring CD4 cells per mil in red there. So they decline a little bit. Then they recover as the virus is cleared. You hit that set point, which you can remain at for many, many years. But the virus is reproducing, and it's killing T cells at high numbers, huge turnovers of T cells. And you may have outbursts of higher virus reproduction over the years, but you can see this number of CD4 T cells slowly decline. We just can't keep up with the killing in the production of new CD4 T cells. And there can be symptoms in this chronic phase. There can be none, or there can be sporadic symptoms. The virus, of course, is evolving in many ways during this time, including in receptor usage, as we've talked about, but it's also evolving antigenically. Antigenic variants are arising in this patient, both B-cell and T-cell variants. And then you enter the AIDS phase, which is defined by the number of CD4 positive T cells per mil, 2 to 500 initially, and then below 200. And there the symptoms begin to arise. You can have various cancers. You can have various opportunistic infections that can't be cleared because the immune system by then is trashed. There are hardly any T cells, the CD8 cells are also going down functionally. The T cells are becoming exhausted, and you cannot clear common infections, and those are what eventually kill you. The opportunistic infections by protozoa, bacteria, fungi, or viruses. So that is the progression of infection. The virus can also infect the gut, of course, by anal sex, or it can be brought to gut tissues by viremia. That leads to disruption of the gastrointestinal epithelia. And so the virus actually infects lymphoid tissue in the gut, the submucosal lymphoid tissues like the Peyer's patches, and that causes inflammation. Right, you have an immune response that leads to inflammation, which disrupts the epithelium, and this allows bacteria in the gut to get into the subepithelial tissues, and that results in even more inflammatory responses, including more T cells which come in, and they, of course, can be infected by the virus and provide more substrate. So this is called Leaky Gut Syndrome, and we think that contributes to the sustained activation of T cells, which the virus really prefers to multiply in. This is a photo that I got from Scott Hammer, actually, who just died not too long ago, and he was the head of infectious diseases at Columbia for many years, involved in many HIV vaccine trials. And this is a disruption of the gut-associated lymphoid tissue after infection. So normal individual, HIV negative. This is a photograph of the intestine, which is lined with lymphoid cell aggregates. So not only are there Peyer's patches below the mucosal layer, but in the intestinal wall themselves there are collections here. These are actually the Peyer's patches. They're very prominent structures, and they're full of T cells, of course. And when HIV infects them, they're gone. This looks like a very nice, clean colon, but it, in fact, is abnormal. There should be lymphoid aggregates there, and they're not. And that's one of the issues that happens. Studies of many infected adults in the absence of antiretroviral therapy, which was done only before antiretrovirals were available, show that approximately 10% of people who are infected progress to AIDS within 2 to 3 years after infection. Over 10 years, about 80% of untreated adults will show evidence of disease progression. Half of them will develop AIDS. And so those can happen quickly. They can happen, in a 10-year period. They can happen more quickly. So there are typical progressors where up to 10 years the virus reproduces until AIDS occurs, and then rapid progressors 2 to 3 years. And then we have what are called non-progressors. Less than 5% of patients remain AIDS-free for many years. They have normal CD4 T cell counts. They're called long-term non-progressors or elite controllers. They do have some virus in them, very low levels, but they control it. And obviously understanding what's happening in them is important. They've been studied for many years. Elite controllers maintain normal CD4 counts, low viral loads for over 10 years in the absence of therapy, 1 in 300 infected persons. So we could certainly learn from them, right? About 20% of them are associated with favorable MHC types, particularly B57 and B27, and also T-cell responses to gag. These are not attenuated viruses. These are not viruses becoming less virulent in the infected person. And you could easily conclude that if you didn't look at it harder. But these people have specific MHCs. And remember, the MHC is presenting the viral peptides to the T-cell receptor on the CTL, and the CTL will then kill the cell. And this virus can change very quickly its T-cell epitopes, that is, those peptides that are displayed on the T-cells. This is something that doesn't change in SARS-CoV-2 variants. That's why we still have high vaccine efficacy, effectiveness against severe disease and death. But for HIV, since the infection is so long, different T cell variants arise in the host, and they can make a difference. The CTL can't recognize the infected cell, but apparently in elite controllers, their MHC is good at recognizing whatever T cell variants the virus throws off. And there are probably a lot of reasons for that, but they're very intensively studied because we want to know if we can translate this to other patients. The dynamics of HIV are astounding. In the absence of drugs, the minimal rate is estimated for release into the blood is on the order of 10 to the 10th particles per day. 10 to the 10th particles per day, with a half-life of about 1 day. So that's 1 cycle per infected cell per day. And that is the engine, high reproduction capacity, that drives viral pathogenesis. Because of the high mutation rate on average, every possible change in the genome at every position is occurring numerous times each day. Every position in the genome is mutated numerous times each day. Think about that. That's a quasi-species. The diversity is astounding, and we're going to take a look at that in a moment. But the virus production, 90% or so from productively infected CD4 lymphocytes. These are activated. The unactivated, they're better if infected when they are activated, which is the process of recognizing antigen and so forth. And then there are other populations that play into this. You have latently infected CD4 lymphocytes, which a few of them can become activated and make virus. You have long-lived cell populations that live for many, many years and constitute the reservoir, and of course that's the problem. What cell populations constitute the reservoir? Most of the studies have focused on CD4 cells. So CD4 cells become, of course, activated when they recognize antigen. The activated cells then can become infected. If they are not killed by HIV, they will become latently infected, and they can persist for some time in the body, and they will periodically be reactivated and produce virus. Most of these cells reside in lymph nodes and lymphoid organs, and again, many of these are memory cells that result after antigen activation. And these memory cells in the lymphoid organs can periodically be reactivated by stimuli of various sorts, and they can produce more virus. HIV also leads to cancers. 40% of infected individuals develop cancers, and these cancers are different from the kinds of cancers that we talked about being caused by viruses previously, where many viral genes will turn on cell division, and that apparently leads to mutations accumulating in those cells and cancers. Here, for HIV, the cancers are indirect effects of dysregulation of the immune system. For example, the virus is killing the very cells that are important for tumor surveillance. And so you have reduced surveillance, and tumor cells that arise randomly are more likely to propagate. But also contributing are the high levels of cytokines that are produced because the lymphoid cells are always being infected. Lots of cytokines, which, as you know, a subset of those can cause cell division, and continued cell division is a recipe for accumulating mutations that lead to cancers. Furthermore, many of us have oncogenic viruses in us. We have Epstein-Barr virus, we have herpes virus 8, we have human papilloma viruses, which can be activated as a consequence because the immune surveillance is reduced, and they can lead to the induction of tumors, transformed cells, which will then reproduce in the presence of cytokines and cause cancers. Kaposi's sarcoma is one such cancer, first described as far back as 1872 by Moritz Kaposi, a Hungarian physician, and it was seen in mainly older Mediterranean men who were likely immunosuppressed, not infected with HIV, right? This is pre-HIV, but immunosuppressed older Mediterranean men. And it would manifest as skin tumors, as shown here, and also their internal organs can be infected as well. It occurs in about 20% of HIV-infected homosexual men, 2% of HIV-infected women, and transfusion recipients. And it's not clear to me why this is different. There's a more aggressive form in certain parts of the world where the virus not only causes transformation of skin cells, but internal organs as well. Interestingly, the tumor depends on another virus, human herpes virus 8, which was actually discovered at Columbia University a number of years ago by Chang and Moore. And you need to have both viruses present to cause the sarcoma. You have to have both HHV8 and HIV. HHV alone is not needed. It does not do it. You need HIV or immunosuppression. So that may explain why these older Mediterranean men had capaces. They had HHV8 and then got immunosuppressed. Other types of cancers arise. Lymphomas, capaces we just talked about, is a tumor of endothelial cells. Carcinomas. Again, infection of various cell types leads to the production of cytokines. These can cause division of, say, B cells to lead to lymphomas, and that may involve Epstein-Barr virus or not. The cytokines cause proliferation of endothelial cells, which, again, with HHV8 may lead to Kaposi sarcoma. And once these sarcoma cells arise, they aberrantly produce high levels of cytokines which maintain their tumorigenicity. And then epithelial cells also respond to the cytokines, and that can lead to carcinomas, especially if human papillomaviruses are present. So a lot of different tumor cell types. And again, the driving force, cytokines that cause aberrant cell division, continuous for many, many years throughout that intermediate period. The cytokines are always produced because there's a low level of virus, and that's driving cell division and reduced immune surveillance. That to do them together, a recipe for these cancers. Well, let's go back to some more questions here. How does sequence diversity within HIV subtypes compare to diversity within subtypes? My understanding is that the diversity among HIV subtypes is greater than SARS-CoV-2. But I don't have numbers for you. What virus has the most potential for causing the next pandemic? Influenza, H2N2. Some kind of influenza, but I think it might be H2N2, based on the comments of Ron Fouché. What do you think about the person who did genome editing on the CCR5 on 2 girls? Yes, so there was a cowboy scientist in China who convinced the family they were going to have twins, and I think one of the parents was HIV positive, maybe the mother, I don't remember who, so they didn't want them to be infected. So he did a CCR5 disruption in utero. Well, he took out stem cells, disrupted the CCR5 gene, then put them back in. And I don't remember where the fertilization occurred, probably in vitro. Anyway, the lady gave birth to two children, twins, and they're HIV negative. They're CCR5 null, of course. Whether or not they end up having... All right, so Cam, my mod Cam is texting me. What's wrong? Thank you. Timer. Thank you very much. You don't mind if I go a few minutes over. Anyway, so that's the science. Whether these girls are going to be okay is not known, right? Because they're still young. Maybe they'll have other issues. So that's the ethical issue. You shouldn't do this unless you know that having no CCR5 from birth is an issue or not. Even though we know people exist with it, their genome is different from the 2 girls. It could be an issue. So that was not a good thing to do. It's unethical, and I think he was fired. So what I think of him is you shouldn't have done that. That's unethical. Why did you do that? You wanted attention. You wanted to be the first person to do it. As I said before, ego drives science, but there's a really bad example of that. Not good. Would a successful HIV vaccine have to block infection? That's a great question. So what we think that in those first 2 weeks is when the latent reservoir is established. So you have a window, and it may be, 10 days or 14 days. I don't know exactly. And, getting an exact number is hard, but I think it would have to reduce virus reproduction substantially. Yeah, maybe even be sterilizing. What a good point, and maybe that's hard to get. How long can HIV remain dormant? So these latently infected cells, memory cells, which can live a long time, right, or hematopoietic progenitors, I think they can last for years. In fact, we have a number coming up. When we talk about trials for getting rid of the latent reservoir, I'll give you that number, how long they can remain dormant. So hold on for that. Can we be sure there's no vector roots? There's a difference between a contaminated needle and a contaminated insect. So the mosquito is a flying syringe, right? Well, contaminated blood. Remember, how much virus is in blood? 10 to the 10th, 10 to the 9th per mil, 10 to the 8th per mil. A lot of virus in blood. Mosquito's not going to have a lot of virus because they're going to take a blood meal and then they're going to fly somewhere else. They take a blood meal to lay eggs, and then they come back later and bite someone else. And I don't think they're going to deliver as much virus. I think it's not reproducing in the mosquito. That's quite clear. So most virus transmission in insect vectors, arthropod vectors, needs reproduction in the vector. There are very few cases of mechanical transmission. So this virus does not reproduce in insects, so that's why it doesn't happen. Hello, Animal Party. Good to see you. How can I get a TWIV cover like yours for my phone? Oh, so this thing. So I made this. I don't even remember the company that I did it. I have to look it up. But send me an email and ask me, and I'll send you the link. Yeah. Is the risk of receptive higher than insertion because of more susceptible cells? I believe that's the idea, yes. In the intestine, there are lots of susceptible cells. And I think, I skipped over that, but I have some notes on that. Let me go back to the frequency numbers. Let's see. Oh. Which lecture? Oh, here it is. Yeah, okay. During sexual transmission, HIV enters the body through genital and rectal mucosal surfaces. These sites differ anatomically and immunologically, and such differences affect transmission and dissemination. Both male and female hormones appear to facilitate transmission by stimulating cell-to-cell contact or erosion of the vaginal lining. Although the insertive partner is at low risk, transmission can occur through cells in the lining of the urethral canal of the penis, presumably from infected cells in the cervix or the gastrointestinal mucosa of the infected receptive partner. Uncircumcised males have a twofold increase of infection. Foreskin may have high concentrations of immune cells susceptible to infection. I forgot to tell you all of that. Yeah. So there you go. That's a good question. Does the needle stick depend on the bore gauge size? I don't know. I've not seen studies that have looked at it, but they might have been done. CCR5 with CRISPR. How can this be done? Well, you have to make a vector. The easiest scenario, actually, we're going to talk about this in the last lecture, the last session of the course, gene therapy, okay? Hold that till then. It's a good question. If you have questions about SARS-CoV-2s, hold them for tonight, live stream, 8 p.m. Eastern. Amy and I will answer them. A company I used to work for has HIV gene therapy. Harvest CD4, modify them. Yeah, that's what we're going to talk about in the gene therapy session. That's exactly what is being done. You take out the T cells, modify them, and put them back. And they will be now resistant, so presumably they will propagate, right? And eventually there will be mostly non-susceptible T cells. With therapies making HIV undetectable in the blood and being told the patient cannot transmit as long as on treatment, is virus reproducing or CD4 cells depleting? No. If you keep the antiretrovirals there at the proper levels, the virus is not reproducing. As soon as there's a latently infected cell that spontaneously activates and begins reproducing, the drugs will inhibit those. There will be those spontaneous firings of latently infected cells every day probably, but the antiretrovirals will prevent them from spreading. If you stop taking your drugs within days, your viral loads shoot way up, which shows that there are multiple spontaneous reactivations frequently. Does triple therapy work in the late stage? So I think even then you can start therapy and the patients will recover, but not beyond a certain point. And I don't know exactly what that is, but people who treat AIDS patients would know exactly. B and NK cells are also impacted by HIV infected. They're not directly infected, but the overall immune balance caused by lack of CD4 positive T cells impacts those as well. And Sean says they do, NKs are correlated well based on receptors and HLA type. Is the reason the level of HIV stays stable that the immune system is still capable? So we have CD8 cells that can restrict infected cells, and we have antibodies that are made which can keep the viral loads low, but the virus does evade that periodically, as you'll see in a moment. Now, HIV doesn't activate cell. The cytokines cause the cell division, and it screws up the immune surveillance. That's exactly right. Does HIV tell us something about more and less mutable regions in the genome? Of course, yes. You can look at the glycoproteins and find regions that are conserved, and in fact, we're going to look at that in a moment for broadly neutralizing antibodies. Among the approximately 6,400 mammalian species, how many are known to be reservoirs of some retrovirus? I think most, HIV, no. The HIV homologs are restricted to non-human primates and primates. But certainly everybody has a retrovirus in their genome. But not, it was the father that was positive. Okay, thank you very much. When I told my students about the origin of HIV, they said, unbelieving to me, who eats monkeys? Apparently other monkeys, right? Oh, no, humans. Yeah, that's a cultural thing. You don't know that because you can go to a supermarket, right? Whereas in remote areas of Africa, they can't. So they have bushmeat, monkeys and bats, right? If broadly neutralizing antibodies are good, why are there no clinical trials? Well, they're not vaccines, right? They're passive. And they are being trialed, but you have to figure out how to induce them because just treating people with them is a temporary solution. All right, let's move to the last part. Is a vaccine possible? So, you get that initial viral load in the early infection, then it goes down, which correlates with an increase in antiviral immunity. So obviously the immune response can, in certain circumstances, control infection, but it still persists. Viral persists, and it eventually outstrips immune control. However, we know superinfection occurs less frequently than the initial infection. People who were infected once have a lower frequency of reinfection. So there's something is happening in the immune system, but we don't know what it is, do we, Mr. Jones? One of the things is HIV escapes neutralizing antibodies. We have a purple HIV arising, infecting someone, and eventually, in a couple of weeks, you make purple antibodies, right? But then the virus has changed its coat. It's become an orange spike. These are antigenic variants. All right, so we make orange antibodies, and then the virus changes. This happens continuously throughout that intermediate period. The virus is always changing. So unless you stop the initial infection, you're going to have a hard time limiting this with a vaccine. And so, yeah, a vaccine may have to be almost sterilizing. There have been many, many vaccine trials, and the one that was thought to have shown some promise is RV144, a trial in Thailand of 16,000 adult volunteers. It was a prime boost regimen where the priming was with a canary pox vector, a pox virus vector, bearing the gag, pole, and envelope genes. All right, shot one. And that's called ALVAC HIV. And then the boost was recombinant GP120 protein. Prime boost. 6 prime, 6 boosts. What a trial, getting 16,000 people to come back 12 times. I applaud them for doing that. Now, why Thailand? Because high incidence of HIV infection there. Now, when you do such a trial, you have to counsel people to use condoms and practice safe sex. But the trial doesn't work unless people don't listen to you. So there is some HIV arising in this population, but my gosh, the numbers. We have 74 cases of AIDS diagnosed in the control population that didn't receive the vaccine, and 51 cases in the vaccine population. 51 cases vaccine, 74 placebo. At first glance, this is about a 31% reduction in the vaccine group compared to placebo. 31%, which would never be approved, of course. Remember, for COVID, the FDA said 50% or higher. And so this is bordering on placebo effect. And in fact, depending on how you crunch these numbers statistically, you can say they are or are not significant. In fact, in more recent analyses, this difference has disappeared. So they have all failed. They continued to do trials. They don't really change much. And I think it's frankly a waste of time, and they should try something else. Maybe mRNA vaccine is the solution. Who knows? They should try it. I would try it. mRNA for what? Probably multiple proteins. But here's part of the issue. Evolutionary distances compared with influenza viruses. So this is the envelope gene and the influenza virus hemagglutinin. So in A, we have a tree based on 96 hemagglutinin sequences from human H3N2 viruses. These are worldwide isolates from 1996. So that's the extent of the phylogenetic diversity in the HA, and this is expanded here so you can see it. Now, B is the tree based on 9 clade B sequences from a single individual collected at one time point 6 years post-infection. One time point. This is as much diversity as a year of HA influenza virus in one person. And C is the tree of HIV sequences. These were sampled in 1997 in the DRC from 193 individuals in a single year. This is huge. 193 individuals in a single year. So this is way more diverse than influenza virus. Influenza virus is comparable to SARS-CoV-2, I would say. So to answer someone's earlier question, it's huge with HIV, and this is a problem with a vaccine. Now, there is some hope in the form of broadly neutralizing antibodies. So during an infection, again, you've seen this graph before, initial infection, years after infection. During this long period where low viral levels are observable, the neutralization breadth of the antibodies changes. You have maturation of your antibody response. I didn't put on the timer. Okay. Damn. Well, I'm not answering questions. What am I doing? I'm just losing it here. During this time, the antibody response matures. You have somatic hypermutation and production of antibodies with different specificities. And you have antibodies of increasing neutralization breadth. You can actually pull B cells out of patients and isolate the antibodies produced by them and show that they can broadly neutralize. And here are some of them shown here. This is the glycoprotein spike, the SU and the TM. So those are both schematically shown in the virus particles. There's a transmembrane there. And you can make antibodies against the very top of the molecule. Here's the CD4 binding site here. And so some antibodies block that. But the broadly neutralizing antibodies can bind different parts of the glycoprotein. They can be in the membrane proximal domain, the GP120 core. They can be directed at the top, or even the CD4 binding site. And they will neutralize a broad range of subtypes. Now, that's the nice thing, and a broad range of antigenic variants that arise in any one protein. So we have these antibodies. We have multiple antibodies. We can treat patients with them. They're in trials for treatment. But as you saw for SARS-CoV-2, monoclonals are not the solution. For SARS-CoV-2, of course, it's because they're directed against not conserved regions. But what are you going to do with this? You're going to give these to someone shortly after exposure or risky behavior? I don't think that's the right approach. You have to figure out how to induce these because they're very rare in an infected person. So that's the key. How do you induce these things? And that's what people are working on. So that's one approach, I think, which has to be done because the current vaccine approaches are not working. But then there's this people who are already infected. There are a lot of those. Can we do something with them? And we have a long-lived reservoir. How can we get rid of that in a patient? I didn't tell you I promised that I was going to tell you how long it was, but these can go years. So a long-lived reservoir, essentially, you can never get rid of all of these cells. They have such a long lifetime. You can try treating people with antivirals and broadly neutralizing antibodies, but that doesn't eliminate the reservoir of proviral infected cells. The two other strategies have been proposed and tested. One is called shock and kill. So again, you have proviruses in latently infected, long-lived cells which can live for years. You try and induce them with chemicals. So you could use a histone deacetylase, some of which have been FDA approved. And you can turn on the proviral expression, start the reproduction cycle, and then hit the cells with antivirals. And the idea being you can kill all the infected cells, the latently infected cells. It hasn't worked. It's been tried. It doesn't work. Block and lock is another strategy. Can we permanently inhibit transcription? So in shock and kill, we're turning on transcription and then inhibiting virus by antivirals. Block and lock, can we permanently inhibit transcription? And people are working on ways to do that at the moment. So that remains to be seen if that works. I tend to doubt it. I'm not sure that that's going to really help at all. I want to go back and see if I can get you the number, which I failed to do. Okay. Here we go. Over 90% of viruses come from CD4 cells in the blood, which have a half-life of 1 day. 1 to 7% comes from longer-lived cells in other compartments, okay, with half-lives that can reach 145 days. So you may say, well, that's not very long. But even if you could block synthesis of virus by drug treatment. It would take 3 to 5 years before these longer-lived compartments are free of cells with the potential to produce virus. But unfortunately, the value is an underestimate of 3 to 5 years. So in other words, you could treat people for 3 to 5 years, and you should be able to get rid of this 145-day long compartment, but it doesn't work. So obviously the compartment is even longer lived. You have to get rid of the provirus or lock it, and that's what some of these approaches are aimed at. Okay, so that's the problem. 145 days, half-life, minimum probably longer. You can't just treat with antivirals. It doesn't work. You have to do something else. So that's where we are. And right now we're distracted by COVID, of course. And a lot of people who have been working on HIV/AIDS are not doing it as a consequence. But we need to get back to that, and I hope we do. So let me end with this slide, which I made many years ago. And this is the story. In 1921, SIV went from a chimp to patient 0. Who knows what that was? And that has led to HIV, 75 million infections, 32 million deaths, and counting. It's just amazing. And not amazing because I think it's good. Some people have accused me of being morbid. It's amazing that we missed this for so long. It just simmered from 1920 to 1980. And remember, we detected it first in New York and other big cities. It started in Africa. So does that mean someone made the virus in New York and Los Angeles? Of course not. You get the point. Next time, Monday. Two more sessions left, but folks. The last week of the year, the last 2 sessions of Virology Live. We're going to talk about unusual infectious agents. This is a fun one. Well, they're all fun in my opinion, but you'll really like that. Okay. Why didn't that last slide show up on the live stream? There you go. Boy, there's a big lag. I want to get back to our questions now. And by the way, I want to just wish you, I happen to be, I'm Italian, so I celebrate Christmas. So I actually have a little Christmas tree here on my desk. I tried to put it on the shelf, but it doesn't fit. Anyway, so you can get a different angle view of the incubator, and all this stuff here is what I use to bring you this. So here's my tree. For those of you that celebrate, hope you have a good Christmas. Let's do some questions. I read a lot of research, let me highlight that, saying nanoparticles destroy these viruses. I'm not sure. Nanoparticles are not good for us, so I'm not sure that that's useful. mRNA is supposedly the key. We'll see. I hope so. I hope you're right. Absolutely. Only humans have CD4 and CCR5. No. Other animals have them as well. Absolutely. And we do have a TWEVO coming up in a couple of hours, Nels and me. We're going to talk about SARS-CoV-2 evolution as usual. Tonight's the Q&A at 8 p.m. Eastern. Did anyone do analysis of the genome of the Lisbon patient? I'm not aware of it. I'm not sure it would be informative. One genome is very hard to interpret. I mean, it may have had, the patient may have had CCR5, right? That would be obvious, but I don't know what else. I was watching something interesting about progranulin in C. elegans. I'm not sure I know what you're talking about. I'm sorry. I think Shane Karate is working on an HIV vaccine. I don't know. The Lisbon patient, here's the reference. Exceptional longevity suggests aging as an ultimate goal for HIV care. Oh my gosh. Okay. Ballad of a Thin Man. I do like Bob Dylan. I do like his music, which dates me, of course, right? But I grew up with him. You're welcome, Ian. The complexity of life and its fragility, definitely, but it's pretty durable, except when humans came along, we tend to mess it up. Does this dance of antigenic mutation with antibody occur with latent TB? I'm not aware that it does. I believe it's more in a dormant state. I've heard about a Nature Medicine paper discussing an mRNA vaccine. Yes, we will definitely do it on TWIV. I'll take a look at that for sure. At each cell division, CCR5 must be reestablished. Is it feasible to downregulate docking on the inner membrane? I think it could be, but we know that we just need to CRISPR out the gene, so I think that might be more straightforward, right? But we just have to make sure it's... You can do that in adults, right? You can do that in T cells you remove and put back, and it's that way you get around the issue of it potentially being dangerous if you do it through fetal development. South African scientists probe link between Omicron and untreated HIV. Well, they should look at that. It's been suggested that it may have emerged in an immunosuppressed HIV patient, but I don't think there's any evidence for that. It's also suggestive of emerging from rodents. So these are all interesting ideas. They have to be proven. Can you give several inactivated vaccines as a strategy? It's not a problem. We give many bacterial antigens together, and that is our immune system can handle it. I don't know if you mean if it's for one virus or for multiple ones. Not sure. And yes, the next lecture is the one where the students go vegan. Yes, when you hear about mad cow disease, you won't eat meat. And Vanity Nutrition, I understand, will be here on Monday. That should be fun. Buon Natale. Yeah, you bet. Thank you, Tom, for your contribution. I really appreciate it. And mods, happy holidays. Les, Mr. Ozzycam, Steph, Vanity Nutrition, have a great holiday. We'll be back next week for 2 more sessions, the last 2 of the year. And if you can't make it, they'll be recorded, so check them out. Thank you, Kate, for your contribution. Really appreciate it. Hey, Nels, look who's here. Nels is here, checking. God Yule. See you at 3 p.m. Nice to see you, Nels. I missed you. We haven't done anything since October. It's no good. We have to be more regular in the new year. Thank you, Greg. Really appreciate your contribution. You like the rest of the desk? You can see it again. It's not the widest shot, but you can see all the stuff that I have on the desk. So the laptop is where I'm showing the slides, but then I have, you can't really see, there's a big monitor in front of me where I'm looking at most things. I have this thing here, which is running the streaming software. This is the video switcher. This is the audio interface. This is another laptop for some notes. And in front there is some channel strips. And this is a clock. This is a cool clock, actually. Let me show you. This is it's an interesting clock that looks kind of like a virus. That's why someone gave it to me. I find it hard to tell time on it, though. And yeah, that's a Christmas tree I just brought in. I was wondering about the use of HIV versus AIDS and SARS-CoV-2 versus COVID. If someone gets a positive test, well, technically, you're testing for the virus, right? So you should say, I tested positive for SARS-CoV-2 or I tested positive for HIV, because you may not have COVID or AIDS, right? You may have no symptoms, so that means you don't have the disease. So, but people misuse it. They say, I was positive for AIDS, I was positive for COVID. So, however, you could say an AIDS test, right, inferring that you're looking for the virus. So that's technically what you should do. Thank you, Tia, for your contribution. Really appreciate it. Thank you, Tom, for your contribution. Yes, historical intent. I understand that. We understand also that I can be cranky. I'm sorry. And look, we have Nels fans here as well, which is great. It's time to call Ralph Barrick back. Yeah, sometime in 2022. I kind of wanted to get him near the end, right? Give him his perspective. But we have some interesting people coming in January alone. Thank you, Barb, for your contribution. Appreciate it. Why could mRNA technology be important? Isn't it a fancy way of delivering stuff? Well, this fancy way worked out remarkably well for SARS-CoV-2. Now, it could be that it's just the spike, right, is a good target because the vectored vaccines have worked well also. But I do think that an mRNA delivery gives you sustained antigen production, which is good. And so I think it's worth testing. That's why I think it could be a changer, but it could be wrong. Although they've spent a lot of money, they might as well spend a little more. But there are a lot of people out there that can use it. And John can brag, I was a fellow student with you. I don't know, Nels, you know John? Reportedly, Walter Reed is preparing phase 2 of broad COVID. I don't know the basis of it. I'll look it up. Can't comment. I don't know about it. It's great that all of you virophiles, not virophobes, come here and chill. I'm going a little longer. I'm sorry, mods. If you want to leave, it's okay. I just want to get to the bottom of this. And look, we love the mods. I don't know if you meant mods. You probably did, not models. Mad Cow, you should have seen how science met UK politics. And I heard that it was very tough. And there's some very good stories about that. Yeah. What is the book, Deadly Choices? That's one of Paul Offit's books. It is How the Anti-Vaccine Movement Threatens Us All. It's actually a history of the anti-vaccine movement. It's very well done, and the consequences. And I've had this for quite a while. I'm bringing books down randomly from my office at Columbia, and I'm just sticking them here. Obviously, I'm going to be changing this up. I want to put a lot of virus models here. So like these two. Do you know what these are? You should know. Rhinovirus, poliovirus. These are $30 each. You can get them, I think, at Shapeways. I would buy a lot of them. Now, my daughter asked me this morning, what do you want for your birthday? I should have said I want some more virus models. I didn't think. It's not, but it's both of our fault. We're trying to get a guest for Tweevo, and it fell through, so that's why. But we'll try and do more next year. We'll do one a month, right, Nels? Viromaniacs, that's another one, yeah. I see. You meant it on the basis of the live stream. Got it. Maybe more of a TWIV question. Is the secondary structure of a virus purely determined by the sequence, or there's some dependence on environmental? Well, you can certainly alter the structure with conditions like pH, right? We've seen that low pH completely changes the conformation of the hemagglutinin. So, yes, it can be influenced for sure. I was in the original Virology MOOC on Coursera. Wow. Kira, you're like historic. That was near the beginning of when I started teaching this course at Columbia. I was recording the lectures, and someone there said, why don't you put them on Coursera? And they actually helped me do it. It was a rare show of assistance from them. Although I don't know what department it was. They're very good at helping with that. And I want to do it again. So I will next year. That'll be one of my goals. Grazie per il corso. Buon Natale. Prego. Maybe the mods are models, too, of civility. Yes, that's what I was thinking as well. Although, being from Wisconsin, Nels and I have the same Midwestern accent. Oh, you're putting them all together. You're lumping them, right? My birthday is January 2nd. And I was born in 1953. I'm not ashamed. So I'm going to be 69 years old. My last year before I hit 70. Hey, at least I don't look it, right? I have a senior pass on the train, and they often say, hey, you're not over 65. Can I donate specifically for a virus model? Yes, of course, you could. You could donate, and I will use it to buy one of those others. Yeah, I'd love to have a phage here, right? That would be cool, or some other virus. So it's not just the sequence that can be selected for and passed on. Well, it is the sequence. But what the sequence can do may be selected on, right? So if the sequence can give a protein that can flip at a pH, it will be selected on. But it's not the protein, it's the genome, right? Another January birthday, very good. Excellent. In any given room, there are always people who have the same birthday as you. Love to go through this course properly on Coursera. Yes, so Coursera, you have short lectures and you have quizzes associated and you have discussions. It's much more organized, yeah. Vanity is December 30th. I didn't know that. Happy birthday. Yep. Close to New Year's. That's it. All right. I've kept the mods enough. So let's say goodbye until next time. Does HIV have its own virophage? Not that we know of. But we're going to talk about virophages on Monday. That's one of the topics. They're pretty cool. We're going to clarify some misconceptions. I'll have a fight with Vincent, who has the deadliest day of the year. Deadest? I don't know. You mean like nothing happening? January 1 is often quite dead, right? Well, thank you, Lynn, this is for a virus model. Thank you very much. I will order it today, and then when it comes, I will show it to you and dedicate it to you. Thank you so much. And thanks, everyone, for coming. I think this is a lovely community, 200 or so folks. Before you leave, just give it a thumbs up. Have a great holiday of any sort. We'll be back next week before New Year's, and we can wish you all a Happy New Year then. Meanwhile, chew on that virology. Stay curious. Have a great weekend. We'll see you next week. Bye-bye.